Thank you very much, Rim, and thank you for that introduction, and welcome everybody to this webinar session by Illumina. And we welcome David Ballard from King's College London, who will also share his experiences of next-generation sequencing in forensic genomics. I'll start with an introduction, and then after about 20 minutes or so, I will hand over to David to continue. Now, the next-generation sequencing in forensic genomics initiative at Illumina commenced about 4 years ago. And the reason it started 4 years ago was because up until that point, next-generation sequencing technologies had been somewhat too powerful, somewhat too expensive, and not really working within the right workflow for a forensic environment. The potential of next-generation sequencing in forensic genomics had been discussed for many years previous to that, but without the technology really being able to fit within the normal workflow within a forensic environment, the opportunity to exploit that was somewhat limited. Now, about 4 years ago, benchtop next-generation sequencing instrumentation, including the MiSeq system from Illumina, became available. And it's at that point that we really started to be able to see how this technology could be employed within a forensic environment. Since that time, we've worked very hard on developing not just chemistry, not just instrumentation, and not just software, but a whole consolidated, integrated workflow package that will start to resemble the typical forensic genomics workflow that you see in a forensic laboratory. Now, I'm sure probably everybody on the phone right now understands this workflow inside out, upside down, and back to front, because this is the workflow that pretty much unchanged has been used in forensic laboratories for more than the last 15 years. And any new technology has to be able to fit and resemble this type of workflow in order to be something that laboratories will even consider as an opportunity moving forward. So a very big part of the Illumina initiative has been taking this workflow and looking at the areas where we can improve on the current systems. Now, these systems have been used to create more than 49 databases worldwide, containing more than 50 million DNA profiles. But we know through the work we've done that there are still samples we struggle with. There are still types of evidence that are difficult to analyze and interpret. And if there's a way that we can improve that, then we really want to leverage our technology to be able to try to achieve that. So when we were looking at how we should address and how we should work on a new technology to be able to leverage in these environments, we looked at some of the more difficult samples that we face. And we looked at the limitations that we face, because we have to understand what we can't do in order to be able to decide what we can do. We see increasing numbers of low-input samples. The advent of trace evidence collection, the use of touch DNA samples in evidence, those types of samples can be very powerful in the context of a case, but naturally, they don't have very much DNA available from which to amplify. If we don't get sufficient material from a particular exhibit, we may not be able to run either the test or the range of tests that we need to get a comprehensive answer and to be able to resolve the case. Mixed samples are probably one of the biggest challenges faced by forensic laboratories, and the forensic community has been looking at all kinds of ways over recent years to try to improve our ability. It's been very difficult to improve what we can do biologically. So in many cases, we've looked at what we can do with software and analysis packages. But if there was a way to improve the biological treatment of mixtures so that from the material we can get more information about what's going on, then the software doesn't need to be as complex. It doesn't need to be as difficult to learn and interpret if we can get more information out of the sample as a whole. And then, of course, there's compromised samples, such as those containing degraded DNA. We know from all of the studies that have been conducted over many, many years that DNA degrades relatively quickly, particularly when it's exposed to the evidence. And if remains or other types of evidence have been exposed to the environment for lengthy periods of time, there may be very little, or in some cases, no full-stretch, long fragments of DNA. And when we think about using STR markers, long DNA fragments are often something we're trying to amplify, and we may not be able to. These samples alone are difficult to handle, but many of the forensic samples that come into the laboratory may fall into all three of these categories. They may be low input, they may be mixtures, and they may be degraded. So any new technology has to focus on that and try and deal with those particular challenges, because all of the other things are relatively simple to do. But if we can solve these, then we can transform the way that samples are analysed in the forensic laboratory. When we think about the important forensic questions we need to answer, there are a number of them. The first is relatively straightforward. Does the sample contain biological material? What is the cellular origin of that biological material? When was that material deposited? What was the age of the donor? Who is the donor of the biological material? And are the crime or the event and the biological material even related at all? These are very logical, very obvious questions that have been asked ever since forensic science has been involved in the criminal justice system. But when we think about the current methods that we utilize, we're only really able to answer one of those questions using the DNA through the workflow that I showed right at the beginning of this session. Now, that's a lot of other questions that we're not very good right now at answering. We have certain types of tests that might tell us it's presumptive, it might be saliva, it might be semen, it might be blood. But it would be nice to be able to be more categoric about that type of decision and also be able to answer questions over and above what we can now. What if we were able to say, no, it's epithelial cells, no, it's definitely this particular body fluid? All of those types of answers would be very valuable in the context of a case. But we can't really do that right now with the technology. If we could find a new technology that could answer more of those questions, that would significantly enhance the role that DNA can play in the criminal justice system beyond that which it already occupies. So, in terms of the tests that are used today, another important factor in the development of a new technology is, can it make the current workflows more efficient? We tend to think of the workflows being efficient because they're relatively quick. We can process forensic samples in anything from 2 hours to 2 days. That's quick. But that's if you're using just one test alone. Not always will one test be sufficient, and not always will that first analysis give you what you need. So if we break down the way that we look at samples today, there are a range of different systems that laboratories use. The top 4 are relatively easy to access through a wide variety of commercial systems. SNPs and mitochondrial DNA tend to be used by a smaller number of laboratories using assays that they've developed themselves. There aren't many out there that can actually allow a wider range of laboratories to use them very easily. If we take routine samples, be they databasing, reference samples, or even routine casework, then generally the first test we use is autosomal STRs because it is the most powerful, and if we get a full profile, it can be very valuable. If we think about degraded samples, we probably still use those same autosomal STRs because there are some short fragments contained within those systems, but we might look at specific mini-STRs. SNPs can be hugely valuable in this sense because SNP amplicons can be made very short. We're not dealing with long repeated stretches that we have to accommodate within the amplicon. We're only looking at single base changes. So we can make them very short, and they're ideal for degraded DNA. In those types of samples where no nuclear DNA remains, the mitochondrial DNA could be very helpful. And so again, it is a slightly different workflow, and it's based on sequencing, not fragment analysis. So again, many labs don't want to do that switch, but it's there as a tool. With sexual assault samples, again, autosomal STRs are very valuable, but we also need to think about YSTRs. They may be helpful in determining low-level male contribution. And then if we look at relationship cases, kinship calculations, those types of analyses, we may look at YSTRs, we may look at XSTRs, and we may look at mitochondrial DNA in addition to the standard autosomal STRs. And again, given that a forensic sample may fall into all of those categories, what the workflow actually looks like is something like this, where you have a battery of tests, a battery of challenges with your sample, and thereby a significant number of decisions you have to make. When you've run all of those tests, you then have to consolidate that report. And so what you can end up doing is an expanding workflow with many tests for many samples. One of the biggest advantages of next-generation sequencing, and it doesn't really matter if you don't know very much about next-generation sequencing at this point, one of the biggest advantages is that you can collapse this workflow into a much simpler and much more streamlined workflow, because you can take all of these types of markers for all of these types of samples and consolidate them into a single test. And that's exactly what we're doing with our forensic workflow. Most of the samples will go through a route that gives you all of the different STRs and a variety of different SNPs simultaneously. And then, if you have those very degraded samples with no nuclear DNA remaining, you can look at mitochondrial DNA, and the workflow and the setup and the preparation is consistent for both. So once you know how to do one of them, you also know how to do the other. All of that is then very easy to report in a single consolidated way. Now, mitochondrial DNA has been used on next-generation sequencing platforms, particularly the MiSeq, for a number of years, while we've been working on a way to look at the traditional forensic markers that many people are more familiar with. And this is just one publication that was put out in about September of last year, which is a collaborative study between a number of groups about high-throughput, massively parallel sequencing, which is the other term we use for next-generation sequencing, because next-generation is probably the wrong word. It's been used for many, many years in a wide variety of applications. It simply means we process many, many hundreds of markers and hundreds of samples simultaneously. So the use of next-generation sequencing in forensics is actually reasonably well established and has been evaluated by a significant number of groups already. And mitochondrial DNA analysis is just probably one of the easiest applications to run on this platform. It's certainly quicker than doing it on CE, and it's cheaper than doing it on CE. But I think where next-generation sequencing had to get to, and where Illumina has been trying to utilize our technology, is to address the question of STRs. Because STRs are the link between current techniques and future techniques. We need to be compatible with all of those databases in order to make sure that any new technology can look to the past whilst also enabling the future. And so that's why we've developed this particular workflow. We've taken the instrumentation, a well-embedded, well-tried and tested platform such as the MiSeq, and we have added to that and refined it such that it's now a forensic version of that instrument that's been validated for and designed for forensic applications. And that's the MiSeq FGX. Upstream from that, we've modeled the type of preparation on the kits that laboratories already use. In sequencing terms, we use library preparation kits, not just amplification, because there are a couple of other things you have to do to make it sequenceable on the instrument, but it's in the same format as you may be used to. Kits containing all of the relevant reagents, everything you need to take your extracted DNA or your FTA punch or your lysate from a buccal swab all the way through to sequencing and onto data analysis. And to do that, we've developed the Forensic DNA Signature Prep Kit. One of the biggest and most scary things that most labs have to deal with when it comes to next-generation sequencing is the analysis, because I'm sure many of you have heard about the large-scale genome projects and just how intensive the data analysis effort is. It's true, for very large-scale projects such as that, it is intensive, and it does require a significant amount of bioinformatics skill. But the types of analysis we need to do, we already know how to do, because everybody's been doing it for years. We just need to wrap that into a piece of software that's easy to use and easy to learn. And that's exactly what we've done with the Forensic Universal Analysis software. So all three of these components come together to sit very nicely into that original forensic workflow that we talked about right at the start. Very briefly, what is library preparation? Effectively, it's 2 rounds of amplification. So it's not using technologies that no one's familiar with. So we simply amplify our loci, be they STRs, be they SNPs. We just amplify them as we do today. The only difference is that on those primers we have a tail. And that tail allows us to do the second phase of the PCR, where we attach what we call adapters. And these allow the amplicons to be sequenced on an Illumina platform. In addition to the adapters, we also add index sequences. These are barcodes, and they allow us to determine which sample is which when we run them on the instrument. It's a molecular label. When we've completed the library preparation process, we essentially have a molecule like this, with our target in the center, whichever locus it is that we're aiming for. We have the tags that have been introduced, we have the index sequences, and we have the adapters. That allows it to bind to the matrix within the instrument and then to be sequenced using sequencing primers. And what is it that we're actually amplifying? Well, this is the content of the new system that we've just released. There are 27 autosomal STRs, 24 YSTRs, and 7 XSTRs. Now, obviously, there are more markers out there than we've been able to put into the system, but it's very important to be aware of that some markers are actually very difficult to sequence, even on CE. So when we try and sequence them using next-generation sequencing, we're only including the markers which work well, because we don't want to compromise the analysis by having markers that work too poorly. Obviously, there are always optimization steps that we need to go through, particularly with a system as large as this. But we have a combination of markers which reflects the international standards for the majority of the countries globally, all in a single system. And on top of that, we have 94 identity SNPs, 24 phenotyping SNPs, which do hair and eye color, and 56 ancestry SNPs. So that makes approximately 230 markers in total. That's a huge amount of information you can pull from one system. There are 2 primer sets, one containing just the identity markers and another containing everything. This is to allow countries who don't yet have the legal ability to process phenotyping markers to do so, because they can simply use the identity systems and ignore the phenotyping markers for now. For those countries that can process phenotyping markers, they would use primer set B for all of the evidential material that came into the laboratory, but they would use primer set A for all of the reference and the databasing samples, because you don't need to store phenotypic information for those types of samples, and there would probably be ethical questions associated with doing so. The amplicon size range goes from 60 to 460, so very similar to the current STR systems that are available, but there are significantly more markers concentrated in the area less than 200 bases, including all the SNPs, to make sure that you can recover information from degraded samples. You can use the input amounts of DNA you're familiar with. The optimum is 1 nanogram, but of course we've gone down significantly lower than that, and you can perform direct amplification, all using the same reagents within the same kit. The kit size is 384 reactions, and you can multiplex up to 32 samples for casework and up to 96 samples for single source. When it comes to the instrument, it's very, very simple. You have 2 workflows: Forensic Genomics, for the system I just talked about, and the research use only workflow, which enables all of the other applications that the MiSeq can conduct, including mitochondrial DNA analysis. And when we put all of that together, the instrument is very simple. You simply create a run using a sample sheet, very much like you would on a CE machine. You get quality metrics that tell you if everything passed or if you need to worry. This is just a positive control showing you that everything has worked. Wherever you see green, it's passed. If anything doesn't pass the threshold, it's orange. It doesn't mean a failure, it just means you have to look at it more carefully. When we look at the data, we can see STRs on the left and we can see SNPs on the right. In the STR, you can see that there's a line across this histogram. This is showing you allele 13, but what it's showing you is that this individual has 2 types of allele 13, and the software will show you the sequence for each allele and where the mutation occurs. That's much more information already than you can take from CE. And when you're looking at phenotyping, you can look at hair color, you can look at eye color, and you can look at biogeographical ancestry. All the major population groups are included, together with subpopulation centroids, which have been trained using 1,000 genomes data. And this just allows you to show your sample, which is in red, where it clusters, and which populations it's most similar to. This is just an example of a sensitivity study. This is another shot from the software. And what it's showing you is a titration from 1 nanogram all the way down to about 8 picograms, so just above 1 cell. And you can see the intensity of each sample increases as you go up through the thresholds. And this orange threshold here basically indicates where you can expect to get a good quality full profile and where you may need to look more closely because your sample is likely to be lower input. It's also likely to be stochastic, and you just need to be careful with the interpretation. But you can see 125 picograms, which is traditionally classed as being the area you would expect a full profile, is well within the comfort zone. And all of the 62.5 picogram titrations are very much achieving or just below that threshold. SNPs are fantastic for degraded DNA. And even though when we look at intact and degraded samples, we see some dropout of the longer STRs, we see fewer STRs for female samples, obviously, because we're not profiling the Y STRs in this system. But wherever the DNA is degraded, we get a full profile of all of the identity SNPs, which give us a discrimination power of very close to a full STR profile, simply from the SNPs alone. So the ability you have to resolve difficult cases where you have compromised material, even if some of those STRs drop out, the SNPs will really give you that power. We have mixtures, which I won't dwell on because David's going to talk about some of those in his talk, and he's got more practical experience of how it can be used in the real world. But the software will show you and highlight where a sample might be a mixture and where you may need to look more carefully. And this is just an example of the SNP results where you see we've got a minor contributor that's around about 5 to 10%, and we can clearly see that contribution in the results. The wonderful thing about sequencing is that you don't just have to look at the length of an allele. You can really see the absolute sequence of those alleles, not only in terms of using them for discrimination, but you can track them through families. So in deficient pedigrees, you can see what the obligate maternal and paternal alleles would need to be. And we can start to estimate things like eye color and hair color. This was a blind trial set of samples that were sent to us. And what we can see is that the estimates that we made using this system allied very closely with the self-declarations that the donors made. It is only an estimation designed to be an investigative lead, but it's much more powerful than an eyewitness report or no leads at all. And everything I've just talked about falls into just this box on the forensic genomic spectrum. Next-generation sequencing on the MiSeq FGX system can also allow all of these other areas of forensic science to be explored, to be exploited, and to benefit forensic science. And what I'm going to do is pass this presentation now over to David so he can talk a little bit more about how we're going to do that. But just before I do so, I just want to take you back to those forensic questions that we talked about at the start. We were able to do this. With the systems that I've talked about today and that David will now take you through, in the future, we should be able to also do that. And that's hugely valuable information that can be generated for the criminal justice system. And so with that, I will just put my acknowledgments up for the people that helped put some of this material together and helped guide this particular introduction. And now I am delighted to be able to turn it over to Dr. David Ballard, who is a senior scientist from the DNA analysis at King's Unit, and he's going to talk about the work that he and his group have been doing using the MiSeq.
Hopefully you should now see, yes, the presentation. Okay, so we are very privileged as a department because we can manage to do research, casework, and teaching all at the same time. We're in King's College London, and we concentrate on both criminal casework for defence and specialist prosecution work. A lot of our work is to do with paternity, immigration, and complex relationship tests. We're an ISO 17025 accredited laboratory, and while our research has traditionally focused on mitochondria and Y chromosomes, more recently it's been focusing more on ancestry, methylation, and next-generation sequencing. Having said that we've focused slightly on ancestry, I'm not going to take any time at all today to talk about the part B of the kit that Nicola was talking about. I'm just going to talk about all the identity and normal STRs that we would use in a normal criminal context. So I'm going to give you some data on some preliminary validation. We're going to have a quick look at some results we've got from criminal proficiency testing and see how they compare with a more traditional analysis. And then we'll go through a couple of cases where I can show you how the use of all of these markers together can help significantly achieve a result which, with the more conventional testing, is much harder to get. So, as far as autosomal STRs go, the kit's got all of the normal ones that you would expect from a European standpoint, so the ones that are in NGM or ESI. What it doesn't have is SE33 at the moment, so I'm led to believe that they are working on including that. Also has all of the CODIS loci, and additionally, it's got the two pentas from the Promega kits, D6, which is a Chinese-focused marker, and then 4 of the mini STRs that were developed at NIST a few years ago. You can see that across all of the chromosomes, you get a nice spread of markers. On chromosome 12, up here, you can see that there are 2 markers that are very close together. That's VWA and D12, which we already know about from previous work, are linked. Also, on chromosome 5, we've got CSF and D5. They're further apart, but they do also share some linkage. However, none of the new ones that are in the kit are linked to any of the existing loci that we already use. And we've already shown that for a criminal context, the linkage doesn't play a role, even if it is useful to an extent in relationship testing. So some of the work I'm going to take you through are looking at the stains that were sent out by the German Stain Commission, the GEDNAP exercise, which is a criminal proficiency test that's run from Germany. Each exercise contains 3 reference samples and 4 mock forensic stains. So we're looking at realistic forensic type scenarios to see how the kit will actually perform. So if we look at one of the stains that was sent in that proficiency test, one of the single source stains, and we look at the balance across the different markers, you can see that this is the y-axis scale shows you the number of reads. So this is the number of individual PCR amplicons that have been sequenced. And you can see that while THO1 is obviously overrepresented, even D1, which looks as if it's very low, for this realistic forensic stain has over 500 reads there, which is easily enough to give a confident answer on what you're going to get. Something to note is that D22, there do seem to be issues with D22 at the moment. The heterozygous balance for D22 doesn't always come out wonderfully, so that is something where for a homozygous threshold you have to increase it. For the other CODIS loci, we get a very similar sort of read count. Once again, all giving us good results. And for the extra markers that are added in, well, the 2 pentas are slightly low. Even they're above 500 reads, and the results you get are very nice. Penta E, because it's so long and has such a wide allele range, once again, you occasionally find that the heavier allele is somewhat imbalanced. But that's, once again, a question of validation and checking exactly what your thresholds are going to be. If we look at all 4 of the different stains that came from the GEDNAP trial this year, you should be able to see that all 4 of them are showing a very similar distribution across all the different markers. THO1, once again, works very well, but in every single case, we got at least 500 reads for every marker and managed to get 4 profiles. How did the profiles match? Well, against ESI 17, which is what we'd done all of our reference and stain samples with, and were able to get complete results. The results from this Forensic NGS kit match totally. If we think about concordance in a more general sense, we've now looked at somewhere between 50 to 100 unrelated individuals, many more if you include related individuals, and we've been able to compare the results we've got from previous results with ESI 17, PowerPlex 16, or Global Filer. And so far, across all those samples and across all those markers, we've only seen 1 discordant genotype. That's at the D19 locus. You can see here that for the 14 allele, we've got over 2,000 reads. The 13 allele, you can see, is the stutter, and then you have a very small 15 allele. if we look at a profile from ESI, you can see something very similar, a 14 homozygous allele. And it's only when you look at a somewhat overamped global filer profile that you see that there's very clearly a 15 allele there as well. We know that global filer has a redundant primer in there to account for a primer binding site mutation in the normal primers that are used. If we go on to the YSTRs, this is your normal Y filer set of markers, minus DYS393, which apparently is giving some problems with amplification or alignment, but once again is something they're looking to bring in, I believe. We also have the extra markers that are in Y23 and an extra 5 markers, again, just to give it a little bit more discrimination. We haven't done much work on the Ys, but what we were able to do is check the results against the Y23 profiles that we got for the GEDNAP samples and for which we've got our certificate back from GEDNAP. And you can see that the full 24 marker YSTR profile for all the male samples matched perfectly between the NGS results and the Y23 results. So once again, the next generation sequencing, or second generation sequencing as Nicola would like to call it, is matching exactly to the capillary electrophoresis results. I'm going to spend even less time on the XSTRs. There were 8 markers that they had originally. They removed 1 because of nomenclature issues. But for the 7 markers which we submitted at the top 2 GEDNAP, we got confirmation that all of the alleles called there matched exactly with what was expected again. So how about the SNPs? We've said that there are 94 different SNPs that are located across all of the chromosomes. We found that for the GEDNAP samples, these are, once again, remember, genuine mock forensic samples. We found that we got full 94 SNP profiles for 14 of them, and for the last sample, we achieved 93 out of 94 SNPs. The one SNP that didn't call was clearly a heterozygote there, but it was below the threshold that we had set to call the genotypes. The mixed DNA sample is also very easy to detect with the SNPs, which has always been a slight worry when using them. So if we look at the heterozygous balance here, this is a single source stain. The way we've shown this is we've just shown how the minor allele is to try to make it easier. So a minor allele of 0.5 would indicate a perfectly balanced heterozygous SNP. And what you can see is the vast majority of them are quite balanced. There's 3 here that are showing significant imbalance. One of these we find is always imbalanced. There seems to be some sort of primer binding site mutation for one of the alleles. But generally, the SNPs we see are good. This is the second single source stain. You can see once again there's one SNP that has an imbalance, but most of them are very nicely balanced and very clear heterozygotes. If we move on to the mixed stains, you can immediately see 2 things. First, there are an awful lot more SNPs that are showing as heterozygous, which is not surprising because there's multiple samples in there, so there's much more chance of getting one of the alternate alleles. And you can also see that virtually none of the SNPs are showing as a balanced heterozygote. They're all very clearly showing as being very imbalanced. So very easy, if you've only got SNPs because it's a degraded sample, to, if you see something like this, start to worry that you might have a mixture. Obviously, the degraded samples, it's problematic anyway. This is mixed stain 2. You see, once again, a very similar picture. Many markers, and a lot of them aren't showing anything like the sort of heterozygous balance you would like. So are these SNPs actually concordant with what we would expect? Well, for the GEDNAP samples, there were no reference results to compare with, so we had no idea. But what we have got is that these 94 SNPs generally come from either the SNP for ID SNPs that were published or the KenKid SNPs that were published. We have some data from Genplex, which was a different application by Applied Biosystems a few years ago. and that did 47 of the SNPs that are present in the Illumina panel. And when we check these historic GenPlex types against some samples that we've run again on the Illumina platform, we find that we get full concordance for all 47 SNPs. For the other SNPs that are there, in confirmed paternity and maternity families, we find we get correct passing on of the alleles as we'd expect. So we don't find any non-paternity issues where we wouldn't expect it. So once again, suggesting that there's certainly nothing immediately obvious wrong with the SNP genotype calls either. So going on to more of the interesting and extra information that we can actually get from this Illumina panel. And most of that has to do with the fact that we not only get all of these markers amplified at the same time, but we do get this allele sequence variation. We find that for a number of markers, there's no significant increase in allele number. So for all of these markers here, generally every allele has the same sequence. So if we look at something like CSF, which is what this is, you can see a whole load of 10 repeats and 11 alleles are just an AGAT repeat motif repeated again and again. To try to make this more visually appealing, in future they're just going to look like this. So I'm just going to replace the motifs that are all the same with a coloured box. So what about the other markers? Well, we find that for a number of markers, we get many more alleles once we start looking at the sequence. So for something like D3 here, we find that we've got an increase in allele number of 100%. So we're getting twice as many alleles with the sequence information as we were before. For something like D12, it's even more impressive. What does this actually mean? Well, if we look at something like D5, you can see all of these are 12 alleles, but about half of them have a different allele structure for the very last repeat. So as far as D5 goes, the normal repeat is AGAT, which is what the purple box represents, but the blue box represents an AGAG repeat. So it's just a single base change in the last repeat, but it does mean that we've now managed to increase the discrimination with a 12 allele. So you could get either one of these two different options, which immediately increases the match probability if you know what the sequence information is. For some of the other markers, it's even more impressive. This is the 20 allele at D2. You can see there are 3 different versions here, depending on how the first set of green, dark green repeats, how many of them there are compared to the set of light green repeats. But it doesn't just stop there. We also have another set of 20 alleles where there's a variant repeat in the middle. So we've now got 4 different types of 20 allele. And then we get even another one. So we get a 5th type of 20 allele, which has a different variant repeat towards the end. So with all of this data, it means that we're starting to get much more discrimination out than we would with a normal CE type output. So the sequence variation causes increased discrimination, better mixture analysis, which is what I'm going to show you in a minute. And it also causes obvious nomenclature issues, because if we think about all of these different 20 alleles here, we have to work out some way of telling between them and how we're going to do that. So this is an example of the first mixed stain from that same German stain trial. What you can see is that it looks like a male-female mixture, and it looks like a 2-person mixture. It looks quite balanced for a 2-person mixture, but we don't know exactly. If we start to look at the Illumina results, we can see that here, for example, at D2, already we're starting to see that of the 3 different alleles that we get, the 11 and the 12 allele are 1 repeat type, a TCTA repeat type the whole way through, while as allele 14 has a TCTA repeat for most of the way, but it also has a TTTA repeat at the end. If we look at VWA, we can see that it's got a 14, 15, and 18 allele. Here now we've got 3 different types of repeat motif. Because VWA is very complicated, rather than writing out the whole motif, I've just called it type 1, type 2, and type 3. But you can see we've got 3 different types, and if you look at the 14 allele, for example, you can see there that it's mainly green, which is a type 1 repeat, but it has a small amount of type 3 at the bottom, which is the stutter from the 15 allele. So immediately you're able to start picking out in that 14 allele the component that's come from one individual, or one allele at least, and the part that's come as the stutter of the 15. In complex mixtures where you're trying to deconvolute them, and proportions are very important, this can be crucial. What's more though, if we look at one of the reference profiles in that same GEDNAP trial, you can see that one of the reference profiles is a 15 homozygote. This would fit in very nicely to the VWA mixture, but if we look at its actual repeat sequence, we can see that this 15 homozygote that's just looking like one peak from our ESI run is actually shown as 2 completely different repeat motifs, and very nicely you can see the stutter also has 2 different repeat motifs. And if we look at our 15 in the mixture, we can see it only as a type 3, whereas if this person was in there, it would have to have both a type 3 and a type 2. So not only is this increased sequence variation helping us as we're trying to look at things like particular proportions of alleles, but we're also able much better to exclude people from mixtures, which as the mixtures get more complicated, becomes much more important. For D12, you can see there's 4 different types. So the 17.3, the 20, the 23, and the 25 all have 4 different repeat motifs. And if we look at D21, you can very nicely see that for the 29 allele, we have both one type of allele at the bottom, a different type of allele at the top, and in the middle we can very clearly see the stutter from the 30 allele here. So all of the sequence variation is helping us as we're looking at these mixtures. I'm going to very quickly show you now some results from the NIST complex mixture trial that the forensic regulator sent out of the UK last year. I'm just going to use one of the mixtures. So this was a complex mixture with at least 2 major and 1 minor component. And what you can see here is that in an even more complex mixture, the sequence variation is even more important. So here we can very clearly see there's a small 16 allele with a larger 16 allele. For D5, we can again see that an apparent homozygote is actually 2 different alleles. Here for D8, once again, we see exactly the same thing, a second allele here, and we can see the stutter there, probably. And even more importantly, we can start to pull out the minor component. So it's something like D13 here. We can very clearly see there's a minor component from the minor contributor in that 12 allele. And if we look at D1, once again, in the 16.3, which is quite low anyway, we can see that, once again, there's a small minor contributor that is also contributing to that 16.3 allele. So as far as mixture analysis goes, it's very exciting, and we're doing some more work on that at the moment, but it should make a significant difference in mixture analysis in the future. And there's one more. So D3, once again, you can see this minor allele in the 17. So for the last 5 minutes, what I'm going to take you through are a couple of identification cases, or in these cases, complex relationship cases, where the whole magnitude of all these different markers played a significant role in being able to solve them. So we get an awful lot of Hall-Sib cases. We're doing a lot of immigration work. This particular one was a test on 2 individuals, 1 male, 1 female. All the parents are dead, but they believe that they may have shared a father. Now, in this case, all of our normal markers that we would use, so the ESI 17 and the PowerPlex 16, which is 23 markers, gave a likelihood ratio that they were more likely to be half-SIBs versus unrelated of less than 3, which essentially means that we just don't know what's going on. We then added the rest of the markers that we have accredited in our lab, which is CS7 from Promega, HDplex from QIAGEN, and 7 of the NIST mini loci that we have as a sort of in-house set. We got 44 STRs in total, and still you can see that at 1.34 the likelihood ratio is completely unknown. Well, we don't know whether they're half-SIBs or unrelated. If we add the extra 5 STRs that we haven't already tested from the Illumina panel, it makes not very much difference at all because we'd already tested most of them, so it just goes up to 1.41. But as soon as we add the 94 SNPs, we can see very clearly that we're now starting to get some sort of more consistent answer. So it's now 30 times against, so it's 30 times more likely that they're unrelated than they are half-SIBs. I'm not going to talk about the linkage unless anyone has any specific questions, but when we were looking at the STR markers to start with, and we said that many of them were linked, obviously by the time you add the 94 SNPs on top, you do start to see some linkage, and because we've got some extra STRs as well that aren't in the MiSeq kit, those are also linked, and by the time we take linkage into account, we find that we can increase that unrelated likelihood ratio up to around about 50. So at the end, with 143 markers, we're now showing about 50 times more likely to be unrelated than related. The second and last case I'm going to show you is a multiple exclusion case. So this was a paternity trio tested by 2 different accredited forensic laboratories. With identifier, there were 3 inconsistencies, all were single step. 2 were paternal, and 1 could either be paternal or maternal. Both reported the results as more likely grandparent or uncle rather than father, which is not surprising. The family insisted it was impossible and wanted a 3rd test done. So we tested a further 26 STRs, so they would have been the same ones as before, the CS7, the HDplex, the NIST. But we could produce no new exclusions. The likelihood ratio was inconclusive on a relationship, 1.2 times an uncle versus a father. So we ran these samples through the MiSeq, and if we look at the different exclusions, what we can find is that for the CSF exclusion, the father was a 10-13, the mother was a 12-14, and the child was a 12 homozygote. Very clearly the 12 has come from the mother, and the 13, more than likely, if he is the father, has mutated into a 12. The D5, we see we've got an 11 and a 13, the 11 showing this extra different repeat at the end. The mother is a 10 homozygote, both the same with this extra repeat. The child is a 10-12, so the 10 has obviously come from the mother, the 12, while it could, without knowing the sequence, have mutated from this 11, because of the particular sequence of this 11, we know that if he is the father, it's mutated from this 13. And finally, we see the D8 exclusion, which is the one that could have come from the mother or the father. Here we can see that the 16 from the child could have come from the mother or the father, and either of these 14s could have mutated into the 15. If we look at the particular sequence of the 16, what we can see is that because of the sequence, this versus this, it's very clear that the 16 in the child has come from the mother. And so then, if we look at the 15 allele, it's very clear that if he is the father, either this 14 or the 16 have mutated to the 15. So we now know that the D8 exclusion is paternal from the sequence. But what we also know is that from all the other sequencing from all of the other STRs, every time a allele was in common between the putative father and the child, not only was the allele length in common, but the allele sequence was in common too. All of the Y's match, which isn't a surprise if the alternative hypothesis would be that he's an uncle rather than the father. All of the X's matched, but since it was a daughter, that only proved that the mother was the mother, which we knew anyway. Also, on top of that, all 94 of the different SNPs matched. So by the time we've gone through all of this, what we're now seeing is a likelihood ratio of more than 110 times that he really is a parent with 3 exclusions as opposed to an uncle. So once again, a very interesting case. So in conclusion, we're getting large amounts of data from one reaction, the different situations more situated to the different types of markers, and I haven't gone into any degraded DNA, but certainly we are seeing lots of uses. I could have shown you a nice relationship case where the X's proved very useful. The genotypes we've found so far are reliable and concordant. I hope that I've shown that the added sequence data for the STRs should improve the mixture analysis. And as we've shown with these complex relationships, it should be good for the complex relationship cases. So what are we doing in the future? Well, we're already looking at the mitochondrial DNA on the Illumina, but we're not doing any casework with it. It's just full sequencing. Looking at the results they've got from Innsbruck, we may well end up looking at using that for casework, but we're waiting to see. One of the things, we've had a MiSeq for a year and a half now, and what we have been doing is putting bespoke SNP panels on there for ancestry, which are working very well. We're looking at doing full concordance and mixture studies, which should be done by the middle of summer, and potentially putting it in for accreditation in 2016, depending on how all the validation work goes before then. So finally, I'd like to acknowledge Laurence, who has done most of the hard work here in running the samples, Denise, who's the head of our lab, and especially Jen from Illumina Technical Support, who proved invaluable in the first few months as we were working our way through how to do everything. So thank you very much.
Thanks very much, David. I hope everybody can hear me. And we are going to take some live Q&A. So could I just request, if you do have a question you would like to raise for either David or myself, then please type them into the Q&A section of your interface. We do already have one question, which I will go through, and it's what about the assay cost and factors that affect that? Of course, that's a very important question, because when we're looking at budgets in the laboratory, we do need to know what the cost per test is going to work out at. So what we've tried to do with the development of the system as a whole is to try and bring the cost per test as much as we can into the range of the current systems that are being used. So it does very much depend on whether you are running multiple samples per run or whether you're doing small batches. The throughput that we recommend for evidence material is a maximum of 32, and for single-cell samples, it's a maximum of 96. If you're running those kinds of numbers through each run, then what you're going to be looking at is somewhere between 40 and 75 euros per sample. So obviously 40 euros there or thereabouts if you're doing 96 samples, up to approximately 75 to 80 euros per sample if you're doing 32 samples for evidence material. Now, obviously, that is a little more than the current systems, but factoring into that, you're getting 230 markers worth of information as opposed to somewhere between 16 and 25 that you are likely to get from the current system. So when you're combining all of that, hopefully that brings it into a range that is realistic for forensic laboratories, particularly bearing in mind that the most likely adoption of the platforms and the technologies is for difficult casework. As David very eloquently explained, there are types of samples that are difficult to resolve using current systems, but you can resolve them when you start bringing next-generation sequencing into the equation. So we also factor in the value of that result as opposed to just being the cost. If you do run smaller numbers of samples per run, then obviously that will become more expensive because the cost of the run itself, there is a fixed cost of the sequencing reagents that you use, and you use the same amount of reagents if you're processing one sample or if you're processing 96. So we do recommend that. But if you run the numbers that we're talking about, then the 40 to 80 euro range is what you would be looking at. So I hope that answered that particular question. There's also another question which regards the markers that aren't included in the system. I think that's always one that is a very real question to answer. When you start sequencing markers, we know historically from the Sanger sequencing that's been done on some of these markers that it is sometimes very difficult to sequence them even using Sanger sequencing. And so you would anticipate that the markers that are difficult to sequence with Sanger sequencing are also difficult to sequence with next generation sequencing. One of the main markers that isn't in the system, as David rightly mentioned, is SE33. It was in the system up until last summer, but then we had to take it out because it wasn't performing reliably. But obviously this is an important marker in certain parts of the world, particularly in Central Europe and Germany, Austria and Switzerland. And so we haven't given up on this marker, but we do need to understand where the issues lie. We know it's very difficult to sequence with Sanger, so we just need to figure out a way to put that right. Similarly, there are some YSTR markers and the XSTR marker that David mentioned that aren't included at this point, but which, again, we would like to continue work on so that we can include those additional markers and improve performance, if necessary, of any of the other markers that are in the system, so that we can evolve this system very much like the current systems have evolved over recent years. This is very much what we consider to be a starting point, and we do look to develop it, enhance it, make sure it's as robust as it possibly can be, and make sure that the software is capable of meeting the requirements that laboratories may have. Next question is, when is this technique expected to be validated, i.e. applicable for courts? Well, as David mentioned, King's College have already started some preliminary validation studies, and there is some additional validation that's going to be conducted during the course of the next few months. There are a number of laboratories who are already engaged with us who are talking about validating during the course of this year and having next-generation sequencing on an Illumina platform in use in casework by the end of this year. And Peter de Knife, who has been a long-term advocate of next-generation sequencing for forensic analysis, he's developing a system that he anticipates will also be in use in forensic laboratories later on this year. So I think over the course of the next few months, you will find that there will be more conversations, more presentations, and more discussions around exactly how laboratories are validating this and when it will be coming into operational use. We also know that there is a next-generation sequencing case going through the Boston courts right now, which is the use of whole genome sequencing to resolve a situation of identical twins. So although that's not the exact same system that we've talked about today, it's based on Illumina's technology, Illumina's chemistry, and that could well be the first one that actually goes to a live trial in the very near future. In terms of cost, there's another question about mitochondrial DNA. Mitochondrial DNA is actually less expensive than the numbers that I quoted earlier for the DNA signature workflow. With mitochondrial DNA, we use an alternative Illumina chemistry, which is one of our regular library preparation kits called Mixtera XT. And so if you're looking at those particular types of samples, I don't have the exact numbers off the top of my head, but I do have a small calculator that can do that. But you're probably looking at somewhere in and around the 25 to 30 euro per sample range for mitochondrial DNA analysis. Obviously, the costs that I've quoted are literally just the Illumina section of the workflow. So you'd obviously have to think about what the cost of the upstream and any other additional processing you would be conducting, but that's the cost of the Illumina components for the workflow. And that, I think, actually brings us to the end of the questions that have been submitted through our interface. If anybody does have any additional questions, then I am more than happy to take those via email. My email address is n for Nicola, Oldroyd, OLDROYD @illumina.com. So please feel free to send me questions. Again, that's Oldroyd @illumina.com. There is one more question that I'll take before we close this out, and that's, are there any ancestry studies in the future? Well, as you saw from the introduction, we do have ancestry estimation algorithms within the software through the SNPs that have been included. David's team haven't done any work on that section of it yet, but we've certainly done a considerable amount of work on that during the development of the system, and we do have groups who are looking at the efficiency of the estimations. Obviously, population studies will also need to be conducted on the systems before they're ready to go live, and so again, we anticipate over the next few months that different population studies will be conducted. And, of course, we will be making all of our developmental validation information available. And, of course, we expect publication on those population results in due course. So I think it's a case of watch this space. The system was only released just over a month ago. We're already very excited at the interest that we've received. There are several laboratories looking to adopt this solution, and as you've seen from David's information, there's a lot of value to be gained from being able to look at samples in a slightly different way and be able to gain more information out of that. So I appreciate that we're now up on our hour time slot, so it just goes from me to say thank you on behalf of Illumina and also on behalf of David and the team at King's for joining us this afternoon. We're very grateful for the time you've spent with us, and again, if you do have any questions, then please feel free to reach out to either of myself or David, and we'd be more than happy to answer those questions. With that, I will say thank you very much.